He also was like the enthusiastic kid, but then he always used to get good result. We would be thinking, will he get the result or not? But he used to get the good, crisp bands. All the results used to be amazing, and he has done a good job. Yeah, over to.
Good afternoon. Can everyone hear me? Okay. Good afternoon. My name is Agastya. I go to Indus International, and I'm going to be presenting the anti-inflammatory effect of play by measuring COX-2 gene expression in PMA and 5FU induced UC-MSCs. Now, I know that seems like a lot, so I'm going to try to explain the main crux of my project very simply. So my project deals with wound healing, which is an intricate process of tissue repair and regeneration that contains 4 overlapping phases. The first phase, as you can see, is vascular response, which is either vasoconstriction or vasodilation of the blood vessels right underneath the skin where a wound occurs. The second stage is inflammation, which is the body's natural immune response to an external stimuli such as a wound. The third stage is proliferation, which is the rapid production of cells such as skin cells and scar formation. And the fourth and final stage is scar remodeling, and this is normally a process that everyone goes through when you can see a scab that is formed eventually fall off and a fresh layer of skin is revealed underneath that above the wound. So we've noticed that these processes are delayed in patients undergoing chemo or radiotherapy and in cases of chronic wound conditions. Now, the main reason that these processes are delayed is due to prolonged inflammation, which occurs in the second stage. And I'm just going to quickly tell you why prolonged inflammation actually happens. Next slide, please. Yeah. So, prolonged inflammation is a result of excessive infiltration of pro-inflammatory cytokines. Now, cytokines are small messenger proteins that essentially go to other cells and they tell them to carry out their functions. So pro-inflammatory cytokines are these messenger proteins that increase inflammation. So this, obviously, there are many different types of pro-inflammatory cytokines, such as interleukin-1, interleukin-6, and tumor necrosis factors that all amplify the inflammation process. And this essentially contributes to the production of cyclooxygenase 2. Now, COX-2 is a pro-inflammatory enzyme that is involved in the conversion of arachidonic acid to prostaglandin H2 or PGE2. And PGE2 is a precursor for prostaglandin that acts as a mediator for inflammation and signs such as swelling, pain, and redness. So basically, just to summarize that, when you have increase in pro-inflammatory cytokines, there's an increase in COX-2 expression, which can be, I don't know how to work this pen. Never mind. So you can see it over here. When you have an increase in pro-inflammatory cytokines, you have an increase in COX-2 expression as well, which results in an increase in PGE2. And all of this results in an increase in inflammation and prolonged wound closure or prolonged inflammation. However, the whole point of my research paper was to find a way to combat this. And the hypothesis was to basically assess the anti-inflammatory properties of play, which has been discussed in the past 2 studies, in downregulating the COX-2 expression in PMA and 5FU induced UC-MSCs. Now, 5FU is one of the most commonly used chemotherapy drugs that's used to cure neck, head, and breast cancers. And we further wanted to investigate if 5FU induces COX-2 expression and the effect of play in downregulating this expression and confirming its role in anti-inflammation. Next slide, please. So the method that we used, we essentially collected and isolated UC-MSCs and cultured them in a fetal bovine serum, 10% fetal bovine serum solution. We then subjected these cells to drug treatment where under varying conditions, which can be seen above. You can see with PMA plus play. So varying conditions, we subjected them to drug treatment. We then extracted RNA using the Trizol method, and we synthesized DNA for semi-quantitative PCR, where we then identified COX-2 and GAPDH genes, which we quantified and analyzed using gel electrophoresis and image software. So now I'm going to talk about the results that I obtained. So essentially, in the first portion, we wanted to test the effect of play on UC-MSCs at P5 and P7 COX-2 gene expression, which was induced by PMA. So PMA is basically a tumor-inducing solution which can increase that. So under in vitro conditions, we identified that the expression of COX-2 was successfully induced using PMA. Cells treated, which can be seen over here, so the higher bars obviously showcase increased COX-2 levels. Cells treated with PMA and play, however, was able to downregulate the induced COX-2 levels significantly to a 0.6-fold in P5 and a 0.3-fold in P7. So this showcased positive results for PMA. However, on the second next question, yeah. So then we also wanted to investigate the effect of play on UC-MSCs at P5 and P7 in COX-2 gene expression induced by 5FU. However, the 5FU cells did not really demonstrate substantial elevation of COX-2 gene expression, which can be seen by the bars. They're relatively similar. This basically means that COX-2 wasn't induced because of 5FU, and thus the results that were obtained were inconclusive, and we didn't really find any form of significance between them. However, this could be an area for further studies which can be carried out. Yeah. In conclusion, our results showcase that significant induction of COX-2 gene expression was present in P5 and P7 UC-MSCs when treated with PMA, and this basically mimics the in vitro conditions for inflammation in our study. Apart from that, we also were able to showcase that COX-2 levels were excessively induced using PMA, and they were also brought down back to its basal levels in untreated groups in both P5 and P7, and with P5 being way more effective in downregulating the COX-2 levels. So according to our results, we can infer that play does have an anti-inflammatory property and can hopefully contribute better to one of the side effects of chemotherapy drugs. Thank you very much.